In this presentation, we're going to be looking at chi-squared, which is one of the statistical tests that's listed on the A-level specification. There are a number of terminologies that you have to take the time to memorize in order to pass any question related to statistics on the A-level biology course. Please take the time to go and learn these off by heart before going any further through the presentation, or you're going to find it a little bit more difficult to keep up with things as we start going into some of the mathematics. There are two main types of hypothesis. There's the null and there's the alternative. In the null hypothesis, you state that there is no relationship between the variables you're investigating, whereas in the alternative hypothesis, that's the more traditional hypothesis that you would have been used to from GCSE. We also can talk about a one-tailed and a two-tailed hypothesis. These don't come up in the A-level biology course very often, but it's worth mentioning just so you're not surprised if the terminologies do. And it simply means that if you have a one-tailed hypothesis, you are talking about a directional hypothesis. This means that you're stating what happens to one variable and the impact it has on the other. So as A increases, B increases, or as A increases, B decreases. The other version of that is the two-tailed hypothesis, which is a non-directional one, where you're saying that there is a difference, but you're not saying if it increases or decreases. So A will affect the outcome of B, but you're not stating in which direction. The next two terms will come up quite often during the rest of the presentation, so make sure you're familiar with them. The critical value is the value on what's called a probability table, and it's essentially the value that you determine the level of significance at. Again, that'll make more sense as you go through. The observed value simply refers to the value that you calculate in your statistical test. A little extension task is at the bottom and simply asks you what that equation is, and that is the equation to calculate chi-squared. So let's go for a little hypothetical. Imagine we're playing a game of chance. It's a simple coin toss, and every time we flip a coin, if it comes up that you win, you're given £100. If when we toss the coin I win, you have to give me £100. Now if we flip that coin 100 times, how many times would you have to lose? So how much money would you give me before you'd start to think the coin is fixed? For example, if it was 49.51 and you owed me £100, would you think the coin is fixed? What about 52, 55, 58, 60, 62? At what point would you determine the coin is fixed? In this particular example, you pick tails, we do the 100 coin flips, and it comes up 60 times heads, 40 times tails. You now owe me £1,000. Was the coin fixed, or was it just really down to bad luck? And this is where statistical tests come in, because they allow us to check for variations in results to see if it was pure random chance that caused them. In particular, the one we're going to look at is the difference between observed and expected values, known as chi-squared. In the A-level exams, there is a common question that seems to be appearing, which asks you to identify which statistical test you would use in a given scenario and the reasons why you would use it. What you can see in front of you now is the three different statistical tests that you're expected to be able to use and the statements of why you would use each particular one. In this presentation, we're focused on chi-squared. So when we use a chi-squared, it is to compare the difference between the observed and expected values using categoric data. You need to write that word for word in the exam as the reason why you're using that particular test. Earlier we mentioned a null hypothesis. In statistics, a null hypothesis is a statement that claims that there is no statistically significant difference or no statistically significant correlation between variables. In the case of chi-squared, we are looking at difference, not correlation. So in the case of a chi-squared, we would say there's no statistically significant difference between the observed frequency of. We'd have to then state whatever was in the A-level question and use that in place of x. And again, we'd compare that to the expected frequency. When we go through some examples, that'll make a little bit more sense. We also have the alternative hypothesis. Generally speaking, this is not something that's asked in A-level exams, but it simply is a statement where we replace that there is no with there is a statistically significant difference or correlation between the values. We also talk about a chi-squared value that exceeds a critical value at p of 0.05. Again, it will make more sense when we work through some examples, but in chi-squared, we are looking for our calculated chi-squared number to be greater than the critical value found on a table at 0.05 level. If we do that, then we can reject the null hypothesis. If we can do that, you're saying that there was some indication that those particular results and the variation in those results was not a result of random chance. The chi-square test is a test of the frequency of individuals in defined categories. What that means is it looks at how many things you find in each different categories in a particular set of data, hence we talk about categoric data. The actual equation itself is written out as chi-squared equals the sum of O minus E squared over E. And we're going to look at some specific calculations later on where we're going to go through those particular numbers. O simply stands for the observed results, so the values that you actually see when you're collecting data from the population, whereas E represents the predicted results, the numbers that you should expect to find based on a particular set of calculations. It is worth noting at this point that I would strongly recommend any student doing A-level biology to take the time to memorize the equations for the statistical tests. The reason I would do that is that some of the specifications say that you have to memorize them, some of them don't make it clear. In 2017, one of the exam boards in the specification said that you did not have to memorize the equation. Unfortunately, that year the students went in, sat the exam, a chi-square question came up, and they were not given the equation in the exam. Now, afterwards, the exam board went back and resolved it by giving everyone the marks or by voting the question. However, when the students were obviously sitting in an exam, when they couldn't answer like 5-6 mark question, it caused a huge amount of stress, it caused a huge amount of loss of confidence, and really put them in a bad frame of mind. So while the actual question was rectified afterwards, it could have had an impact on their other grades. If they had memorized the equation off by heart, they would not have been fazed by that. So my strongest recommendation is memorize the chi-squared test, and I would even suggest to memorize the equations for the other calculations as well. So how do we calculate the expected frequencies? Well, the expected frequencies can be done in one of two ways. We can either look at a standard inheritance pattern and look at the ratios. For example, two heterozygous parents and monohybrid inheritance would give us a 3:1 ratio. Same heterozygous parents, but a dihybrid inheritance would give us a 9:3:1 ratio. So we've got these fixed ratios for certain circumstances. The other way of doing it is that we actually do a Punnett squared to work out all the possible outcomes, and then we use that to calculate our ratios as well. So those are the two main ways, and on the A-level exam, they tend to be leaning more towards you actually doing a Punnett square and working out your ratios from there, but it could be either way. As a very broad statement, it is worth noting that most of the time we would be looking for any variation between observed and expected results to be down to random chance. But read very carefully on the question that they've given you, because in some cases you may want to find a significant difference between the outcomes, or in other cases you may want to find no statistical difference, and therefore it is random chance. So please do read the A-level question carefully. If you're to memorize the chi-squared equation, I would always suggest memorizing it in the structure that it's in this particular table. So O, E, O minus E, O minus E squared, O minus E squared over E. The reason I suggest you lay it out like that is because if you're ever asked to actually do the calculation by hand, this format and this table will mean that you don't make any silly mistakes. It's up to you. You can, of course, use most scientific calculators and punch all this in, and it can do the calculation for you. But it's a kind of win or lose, because if you punch the wrong symbol or number in the calculation, you get the wrong value, you've no working out, you get 0 marks. If, however, you have this layout, you will obviously pick up some of the marks, even if you have made some sort of error in the calculation. On the A-level specification for AQA, it does state that students may be tested on their ability to select and use chi-squared test to test significance and difference between observed and expected results. However, the number of marks that will be allocated to this type of calculation compared to the amount of time it would take this calculation to be done by hand is disproportionate. So what I'd most likely expect is that you'd be given a partially completed table and asked to fill in the remaining few details rather than asked to do it all from scratch. If we go back to the earlier example where we had the coin toss test, and it turns out that you owe me £1,000, we had 60 to 40 in terms of the outcomes. You were asked to think up your own numbers, and what I'd like you to do is, whenever I go through this example, is using your numbers, do the calculation a couple of times and see if you can work out at what point the coin would actually be fixed. In the table, you can see our observed tails were 40 and the observed number of heads was 60. The expected numbers, well, if we flip a coin 100 times and there's 2 sides and everything's equal and random, we should find 50-50 on either side. If we do the calculation of O minus E, we end up with minus 10 and 10. We then square those to get us 100, and then we divide by the original E value, which gives each of them a value of 2. In this particular case, the numbers just happen to be the same for each of the different categories. That's not always going to be the case. But at the end, we add all those values up, and that gives us our chi-squared value. At this point, pause the presentation, put in your observed values, and have a go at the calculation again and see if you can come up with the chi-squared value. Once you've done that, we're going to go on and look at probability tables and seeing about was the variation due to random chance or not. This is an example of a probability table. It's quite a large cross-section of probability tables. It's not the only bit. It goes on further to the right, and it goes on much lower down, but you wouldn't be expected in an exam to use anything beyond this. Most of the time they give you a much shortened down version of this probability table for your particular question. There's only really 2 columns that you need to focus on. The first column is what's called the degrees of freedom. The degrees of freedom is a value that we work out based on how many categories we have in our calculation. So in terms of our coin toss, we only had 2 categories, heads and tails. So the number of categories is 2, minus 1 means we have 1 degree of freedom. In other questions, you may have 4, 5, 10, or 15 possible categories. All we simply do is work out all the different options that we had, minus 1, and that gives us our degrees of freedom. The reason for this is because the more categories we have, the more variation we could have by random chance, so we have to change and adjust our critical values accordingly. The second column we need to focus on is this 5% chance level or probability level, or P is 0.05. What we have to do when we get our chi-squared value is determine if we fall on the right of the 0.05 value or on the left of the 0.05 value. Now, I will go through a particular format for writing down your conclusions to the chi-squared test, and again, I would strongly suggest you stick to that format, because if you stick to the format, you'll cover all the bases and get all the marks. But for now, we're just going to talk about if we accept or reject the null, if our difference is significant or not significant. When we look at the chi-square value that we calculated from our results, we need to see if we're on the right or on the left of the 0.05 probability level, and we said we had 1 degree of freedom. What this means is that we are on the right side of the 0.05 level. We're in the significant null rejected side. So once we've identified that, we then have a particular format that I would recommend using when giving our values and when giving our conclusions. To make it easy for the examiner to follow, I've clearly stated what my chi-squared value is, and immediately after I have identified what the degrees of freedom are for this particular question. Again, because it was heads and tails in terms of outcomes, we only have 2 categories. -1 gives us 1 degree of freedom. The next thing that we state is that our value for chi-squared exceeds the critical value when p is 0.05. That's the first sentence. We always say chi-squared exceeds or is less than the critical value. In this particular example, it exceeds when p is 0.5. The next statement we say is, do we accept or reject the null? Because our value for chi-squared fell on the right of the 0.05 level, it fell into that bigger number, we can say that we reject the null hypothesis. That leads us to be able to say that there is less than 5% probability that the results were due to random chance. Do not say the results were not due to random chance. That is wrong. We are simply saying that the probability that the results happen by random chance is less than 5% and it's so low that we don't think it was random chance. It's not impossible, which is why we have to do repeats of experiments. But what you cannot do is you cannot say that there is no chance that the results were random. The last statement is we say that there is a significant difference between our observed and expected ratios. Again, that brings in our alternative hypothesis, and we're just simply stating that to finish off. So the format is state your chi-squared, state your degrees of freedom, say if your chi-squared is greater than, exceeds, or less than the critical value when p is 0.05. We then accept or reject the null. We are the less than or more than 5% probability results were due to random chance, and we can then say that there is or there is no significant difference between our observed and expected results. I really strongly recommend you stick to that format. If you stick to that format, you will be fine in the A-level exams. If you want to do your own version, it's absolutely fine, folks. You're of course entitled to do that, but you are at risk of missing some important piece of information out, and it can make your answer drop quite a lot of marks very quickly. Just to give you a little practice looking up values on the critical value table, there are 3 questions, A, B, and C. We have all the null and alternative hypothesis and even the chi-squared value calculated for you. Again, this is probably the more likely scenario in the exam where they give you a calculated chi-squared and they ask you to use a probability table and then give the interpretation of the results. If you look at A, B, and C, look at the chi-squared values, look for the degrees of freedom, look for your critical value. Remember, it's always that 0.05% level unless the question states otherwise. We always go with the 0.05% level and simply work out if you have significant or non-significant, what kind of conclusions you'd be able to make. So hopefully at this point you've paused the video and had a chance to have a look through and try and work out the answers yourself. Well, in A, we have 2 categories. We have men and women. So our degrees of freedom would be 2 minus 1, which would give us 1 degree of freedom, which means we're dealing with the top row of numbers. If we look on there at the 0.05% level, our critical value is 3.841. Our chi-squared value is greater than that, which means we are on the right-hand side of our probability table, which means that we would talk about it in terms of significance. In B, we are looking at different categories. But in this particular example, it is nurses and doctors, so it's still 2. Again, 2 minus 1 gives us 1 degree of freedom. So once again, we'd be looking to the top row. We go to our 0.05% value, which is 3.841, and we would look up our chi-squared is lower than that, so we're on the left. That means that we're talking about not significant results. We're talking about things happening by pure random chance. In C, there are 5 different subjects. So it is 5 minus 1 means we have 4 degrees of freedom. So we'd actually move down and would move on and use the 4th row down. And you can see immediately our chi-squared value at 0.05, our critical value at 0.05 jumps up quite significantly. And when we look at our chi-squared observed value of 10.557, we would fall on the right side of the 0.05 level, so we'd fall on the significant side, and we'd go through the same steps from the previous slide to give us our full conclusions. At this point, I would suggest you get the chi-squared worksheet that complements this lesson and have a look through the second part of the page where it goes through task 3 and looks at some specific examples where you have to calculate chi-squared. If you don't have access to that, that is available if you're one of my students for download through our shared network area. And if you're not one of the students, have a look at the comment sections below how you can get your hands on one of those sheets as well as a paper copy of this presentation. First example is an ecology-based example in which you are given data for 4 fish species taken from a particular location. It gives you the observed numbers, but it doesn't give you the expected numbers. The expected numbers are meant to be calculated by assuming that if randomness is occurring, the numbers of the fish inside the area should be evenly distributed and equal in number. So that's how you get your expected values. What you then have to look at is your null hypothesis. In this particular question, the null hypothesis is stated, but you may have to in an exam come up with your own. And again, just use the format that I've already given you. So it would be there's no statistically significant difference, because we're looking at chi-squared, between the observed frequency of the fish species and the expected frequency. It's just using the standard format from earlier in the presentation. Go back if you need to sit and memorize that format. You can, of course, state the alternative hypothesis as well. And again, we're going to look at the chi-squared value and either accept or reject the null depending on what comparison we get the critical value at the 0.05 level. If you're looking at this presentation and you are now looking at your sheet and you don't have a table, something similar to what's in front of you on your paper, because you've done everything in a calculator, that's fine, but as I said, it's a win or lose. I really don't recommend you do it that way, but your choice, your exam. If you have gone through the time and effort of doing the calculation and sketched, it doesn't have to be perfect, just a sketched out rough table, what you should find is when you go through your values, you've got a grand total of 40 organisms. The expected you should have had 10 of each. In each row, we do O minus E, and we get each of our respective values. We square each of those numbers, remembering that will remove any minus values. Then we get each number and we divide it by the value of E. Again, in this particular question, E happens to be the same number for all 4 of the different rows. That does not always have to be the case. For example, in a 9:3:1 ratio, E would be different for each of the rows. Once we've done that, we get a value. We sum those values up to give us our chi-squared value, which in this particular example is 10.2, and we then go to our format and critical value table to see about our conclusions. In this particular example, we have the shortened down version of the critical value table. So it's not giving us all the values, it's literally just giving us the one column of where P is 0.05. Just as in the standard format I've told you to memorize, we start with our value of chi-squared nice and clearly identified for the examiner, which in this case is 10.2. We then state our degrees of freedom, which again, as we had 4 different fish species, it's 4 minus 1, which gives us 3 degrees of freedom. The next statement we say relates to chi-squared and a critical value, and in this case, our value for chi-squared exceeds the critical value for p of 0.5, which is 7.82. That means that we are going to fall on the right of the 0.05 level, which means we fall on the reject the null hypothesis side. That means that there is less than 5% probability the results are due to random chance. Remember, folks, if you say there is no probability the results are due to random chance, you will lose marks. That is wrong. We can only say less than 5% or more than 5%. In this particular case, because we're on the side where we reject the null hypothesis, we simply say there is less than 5% probability. And to finish off, we say that there is a significant difference between our expected and observed frequencies of fish. That means the results were not due to random chance. Example number 2 is a standard dihybrid inheritance using 2 parents that are heterozygous for both conditions and results in a 9:3:1 ratio. This means in order to work out our expected ratios, we get the total population, we divide it by 16, and then we times it by 9, times by 3, times by 3, and times by 1 in order to work out the expected populations for each of the different phenotypes. A classic example of dihybrid inheritance is the use of different grains, and you can see quite clearly in A, B, C, and D in the picture that you have 4 different phenotypes, and it gives you the descriptions of A, B, C, and D in the text. And this is what we use for our dihybrid inheritance. In the question, you were already told it was a 9:3:1 ratio, but another way that you may have had to work that out is literally by doing the Punnett square, looking at the 4 gamete combinations for the 2 parents that are heterozygous for both conditions, and you can see there are 16 possible outcomes, which would give you the 9 phenotypes the same the blue, 3 of the phenotypes the same red, 3 of the phenotypes the same green, and 1 phenotype which was represented by the pink, which is the completely recessive for both of the different genes. Just as before, when we're talking about null hypothesis, we use the standard format, and in this particular case, we would say that there is no statistically significant difference between the observed frequency of the maize grains and the expected frequency, and you can mention the 9:3:1 ratio as part of that if you wish. Again, we can talk about the alternative hypothesis, where we simply replace that there is no with there is a, and exactly the same format as below. And once again, we're going to look at our chi-squared value, compare that to the value in the critical value table where p is 0.05, and see if we fall on the accept or reject the null side of that table. So now we've got a null hypothesis, we need to actually do our calculations. It gives us in the question the observed values. So the expected values, again, we're going with the 9:3:1 ratio. Well, if we have 433 organisms in total, you should find the 9 represents 244, 3 represents 81, and 1 represents 27. For each row, we then do O minus E. We then do the square of that to get rid of the minuses, and then we divide each of the rows by the related expected number on that particular row. So for example, in the first one we're dividing by 244, in the second and third row we're dividing by 81, and the bottom row we divide by 27. We then take those values, add them up together, and that gives us our chi-squared value. Now that we've done the calculation for a chi-squared value, we make it nice and clear for the examiner by stating our value for chi-squared. We then go into our degrees of freedom. In this particular question, there were 4 possible outcomes. So the number of categories is 4. minus 1 gives us 3 degrees of freedom. Then we go into our statements, and again, first thing we do is we make a comparison. So our chi-squared value equals the critical value. When they are equal, we treat it as if it's on the right, as if it's on the reject the null hypothesis side. In the exam, it's incredibly unlikely you will ever get the numbers to be exactly equal. It normally very clearly falls on one side or the other. If we take that equal means we reject the null hypothesis, our second statement, then we can say that there is less than 5% probability that the results were due to random chance. Again, folks, I keep saying it, please don't write that there is no probability or no chance the results were random. That would be wrong. You can only state less than 5% or more than 5%, and in this particular question, we state that there is less than 5% probability because we are on the reject the null hypothesis side. And finally, we finish off with the alternative hypothesis that there is a significant difference between our observed frequency of maze grains and the expected frequency of maze grains. In example 3, we are introduced to the concept called contingency tables. Now, contingency tables are not mentioned on the specification, so if they were to appear on an A-level exam question, you would be given an explanation and you would be given an equation to calculate the expected results similarly to what's on the worksheet. But essentially, contingency tables where we're looking at two different quantities at the same time. So for example, in this question, we're looking at color blindness, and we're looking at color blindness in males and color blindness of females at the same time to make comparisons. In this question, the null hypothesis has not been stated for you, but it's exactly the same format as we've been using all the way through. So there's no statistical significant difference between the observed frequency of color blindness in males and females. Exactly the same format as we've been using all the way through. Again, our alternative hypothesis is that there is, and we can then of course look at the chi-squared value, and we're looking for it to exceed the critical value where p is 0.05, and that means it falls into the reject the null hypothesis side, and we do the relevant statements to match. So contingency tables are not listed on the specification for a number of the A-level courses. This means that the exam board cannot ask you about them and expect you to know them going in. However, they can give you equations and expect you to be able to use them to calculate expected frequencies. So rather than wait until the exam to see this for the first time, this is why it's introduced here. When we look at the calculation, to work out the expected number for any of those combinations of male, female, colorblind, not colorblind, we get the row total multiplied by the column total and divided by n, with n represented by all of the numbers added together. If we look at a couple of examples, so the expected frequency of colorblind and male, well, the row total is the top row, so it's 56 plus 14. The column total is the first column, which is 56 plus 754. And n is the total number of organisms added together, which is 1,360. That would give us an expected value of 42 for the colorblind males. If we looked at another example for the males that are not colorblind, again, we're looking at the row, it's the second row, so it's 754 and 536. We multiply that by the column, which is 56 and 754. And we divide that by the total number of organisms, which is 1,360. And that gives us an expected number of 760. Just to make it easier myself and just to make it easier for the examiner to follow, I would put something like this into tables similar to what you can see, where I've got my observed values clearly labeled and I put my expected values underneath. And I simply go through each of the calculations and produce each of the numbers. And then I go through just like before, O, E, O minus E, in exactly the same format as I have done for all the other. And you'd end up with these values. And then you add them all together, would give you a total chi-squared value of 12.33. I have done a shortened version on this one, but please do feel free to stick to the standard table that we've been using all the way through. This is just a short version so you can check your answer. You should still go through all the stages. Once again, we're using exactly the same format. We start off clearly identifying our chi-squared value for the examiner. We then go on and look at our degrees of freedom. Incidentally, for this particular format, we use a different calculation for degrees of freedom. You would not be expected to know this going into the exam, as I've already said, it's not in the specification, so they would have to give you that calculation as well. And when we do, we get a value of 1. Then we go through and look up our critical value on the table at the 0.05 level, and we look, is our chi-squared greater or smaller? In this particular case, our chi-squared exceeds the critical value table. That means we're on the reject the null hypothesis side, which means that there's less than 5% probability the results are due to random chance, which means that there is a significant difference between the observed frequencies of colored blindness in males and females. What we're saying there is that it was not random chance. The number of males that were colorblind was different to the number of females that was colorblind. And if you go through the presentation on sex determination, you'll see why that's the case.